Kia ora, I'm Aaron Wilton. Thank you for joining us here at the Allan Herbarium today.
Yeah, and I'm Kate Baldwin, so great to be here.
Great. So, Allan Herbarium, what happens here?
Okay, so the Allan Herbarium is a facility where we hold material around our flora. So what we're doing is there's collections of material from around New Zealand and around the world, and our job is really to look after those collections on behalf of New Zealand and to ensure that they're available for scientific research and for other users around the community.
So you've got quite a range of displays of the collections. Can you talk us through some of them?
Yeah, so we've actually got a range of material out, and what we'll try and do actually is bring them a little bit closer so you can see a little bit more of the detail. So that'll be just a little bit, just bear with us while we're doing that. So across the collection, we have a range of material, and I'll start with just showing you some of the diversity that we have. So right down in the front here, we have a little lichen. So common through the environment, trees, rocks, you'll see them all around. One of our little liverworts. So
it's obviously displayed very carefully.
Yep.
Talk us through that process. So you go out and collect, bring them back.
Yep.
What happens?
So the researchers or people from, let's say, DOC or MPI or regional councils, they collect material as part of their work, and when it comes back it's actually dried so that it has a long life, and pressed. Most of it's pressed, unless it's something bulky like this one here, where it just gets dried so that it lasts a long time. Then they get mounted, if they're one of the flat specimens like you see beside me here, they get mounted on this sheet so that they're easy to handle, and it supports the material because they can get brittle. And there's little straps you may be able to see on the specimen that actually hold them in place. And we do that so that the material is preserved, but also so that people coming into net that need to use the material can actually cut this material, or they can cut the straps, or they have to flip it so that they can actually see the features of the plant that they're actually looking at in detail. So they're all mounted, they all look very beautiful, but they're actually mounted to try and make sure that the essential features to understand these species are visible for workers.
So talk us through some of the essential areas of this particular species here.
So these specimens here were collected on Cook's first voyage, so sometime 1769, 1770. So you can actually see how well preserved that material is given its age, and that kind of highlights one of the values of this type of collection in terms of the longevity. It's a long-term piece of infrastructure for the country. So these ones went back on Cook's boat to England, and in the mid-80s a number of specimens were returned to New Zealand, and some came here, some went to Te Papa, and some went up to the Auckland Museum.
We've had a couple of questions. Just explain, where exactly is this facility?
Okay, it's a good question. The Allan Herbarium is about 15 k's out of Christchurch in Lincoln. This is actually not the first place it started, so it's actually moved around the country. It actually, in its history, had a little time in the middle of Christchurch, a time in Wellington, and its very first genesis was up at Palmerston North.
That was in the
1895, roughly, yeah. So 1920s in Wellington, and then it's moved out to this purpose-built facility here in the 1960s.
So, talking Techweek, we're talking about the digital experience and the digitisation of these. So talk us through that process. What is a digitisation expedition?
Right, I'm going to answer that slightly long-windedly and come back to your question if that's all right. So with the material we hold in here, access to it is really important to a whole number of people. So people in DOC, MPI, regional councils, iwi. The material we hold here might represent taxa that are not in the wild. And so we're very conscious that we want to provide access to that information to that range of people and the researchers that need to use it, both in New Zealand and around the world. And so the whole digitisation thing is driven by how we can provide access to this material for people to use it so they know what we've got. And there's really a number of benefits from that. For example, it means that, I would say, researchers can come in, they actually can work out what they need to see, what we hold, which means they may not need to collect again in the field. They might be able to pick which specimens they might want to borrow to look at. So we kind of act like a library with other institutes with the, on a loaning basis. And just even locally, you know, people interested in their area, they can actually come online to our websites and they'll be able to see those holdings that we have, and kind of go, oh, so that was collected, you know, locally and maybe isn't there anymore. So yeah, there's a range of benefits and what's motivating us around actually providing the access to this material.
So talk us through what is involved in the digitalisation of this.
Okay, I'll hand over to Kate in a minute, but one of the things in the herbarium is we rely a lot on volunteers around our work, and so when we wanted to get this expedition up, we had to call on our volunteers to start helping us. So they've actually done a lot of the work in terms of mounting some of this material right from its start, so they're key for us in doing that. When we started doing this digitisation, our volunteers actually do a lot of our imaging work for us. And so in the setup behind, you can see with the camera, we have volunteers that come in every week and will be supporting us with by taking specimens that we need digitised and creating those photographic records. There's a bunch of other stuff in terms of the expedition that we're going to see online, and I'll let Kate talk about what she's had to do to kind of get that up.
Yeah, so part of the information that we're wanting to digitise is in fact the information about what the plant name is, who collected it, when, where it was collected from, and anything else that's happened in the life of the specimen. So usually we do it, we sit down and some of our volunteers or staff will actually enter the data that is on the specimen label, looking a bit like this. But this time we're doing it backwards. We're going to get you guys to do that. So we got our volunteers to actually take the images of the specimens, and then we're going to put them up online for you guys to enter the information in, so then we can push it through to our database.
So that sounds like a really cool opportunity. Talk us through what people would need to do to get to that final stage.
Right, so we'll show you the link to the expedition later. So you had to log on to the website, so it's called Digivol, it's short for Digital Volunteers, and the Australian Museum and the Atlas of Living Australia host this site. So museums and institutions across the world actually use this site and other crowdsourcing sites to digitise their information, not just specimens but field notebooks, and they also have like cameras, that wildlife photos and things like that. Quite cool. Anyway, so you'd have to log in, they get a few details, and then you can find our expedition online. There are a number of them, but our one is the grasses and sedges of Aotearoa, New Zealand. So if you type in grasses, there's about 10 or 11 that pop up, and then you can scroll down to our one. Once you click into it, you start look at tasks. So each image, which sort of will look a bit like this, is a task, and then you look at the image label and enter the information into the fields that were provided. We've also provided a tutorial so that it will say, well, we want this collect information in this field, and this information looks like that. And if you have an issue, tick this box, or there is a forum that you can ask questions on, and then ourselves and one of our volunteers will be answering those.
Out in Banks Peninsula, so this is a new grass that's actually turned up, or was noticed about 5 years ago, and we've worked out what it is, and so this is a new thing that potentially could be a problem. So part of what we're trying to do is keep on top of that, so we know what's coming in and becoming wild. But there's also we don't have a specimen out of it here today, but there's even new grasses being discovered and described in our indigenous flora. So there was a new limestone species just published a couple of years ago in the grasses, for example, and that patterns across the whole flora. So all of the groups that are out there, there's new things being worked out, new relationships.
Kate, did you want to show some of the digitisation?
Yeah, we'll give it a go.
Give it a go.
Let's see. So I don't know if you can see that, but this is the Digivol website. So you come on here, you find the link, and you go, oh right, so I want to get involved. So you create a login, and I am already logged in. That's awesome. So then you go to an expedition. So yeah, there are a number of expeditions online. Slowly, it's thinking. So, because I searched for it before and it's the newest expedition, we can see that our expedition, Grasses and Sedges of Aotearoa, New Zealand, is at the top here. You can also see on the side here is that we've got all these stats about volunteers that are online and have been doing digitisation. Not on our one yet, but on all the other ones. So you can get sort of, there's an honours board and things like that. So, clicking on the expedition. So you know, this is the homepage of it, and so you can view the tutorial or get started. We'll have a quick glance at the tutorial and then we will get started. Tutorial. So we have instructions about how we want these to be transcribed. There's just things about handwriting, about what you need to transcribe, what you don't need to transcribe, what a scientific name is so that you can recognise it. We also have about how we like dates, because we have lots of different handwriting. Most of the older specimens, well, all of the older specimens are handwritten, and there are handwritten notes on the newer specimens as well. So sometimes it can be a bit hard to understand, but we've got instructions about how to do that. We've got georeferences and altitudes, so we can see where, if the collector has said where that specimen is. And we've got all these different collectors and all their different handwritings about what their names were. We've got some suggestions that often grasses, there's descriptive words about grasses. So we've got some suggestions there because they're not often common words, so maybe a bit difficult to spell. And we've got information where you can find things. We've got examples of what some labels will look like and how we would want to transcribe, like what the locality information is, who the collector was, how to work out which name came first, because as the time goes on, the scientists look at it and go, oh, actually it's this specimen instead. So there are often names updated. So we've got all that information as reference, because some of, as you can see, some of the labels actually have quite a bit of information on them.
So is this something that a school could do?
Yes.
Or, yeah.
Yep. So if you can read and you can type and you can use a computer, you should be able to do this. Sometimes it might be helpful to have a adult along as a help, or do it as a group. But yes, you can, and we're always happy to answer questions. And we as part of the Digivol process, we have ourselves or another of our volunteers will be coming along and checking what you do, so validating it. So even if you do make a mistake, that is all right. We will find it. And we make mistakes ourselves, so that's okay.
So we've just had a question come through. This is obviously for species and ones that have already been collected. And
Yes.
What do people do if they think they've found a new plant? What should they do? Can they name it like after themselves? How much do you give them?
Right, Aaron, I'll hand it over to you. You'll sign.
So that's a really interesting question. There is actually an international code on how you name plants and some kind of protocol that's established around it. Increasingly, we're trying to work with iwi and hapu in the naming process of material that have been collected in their rohi. So that's an important part. So the naming of it actually, for scientific purposes, needs a Latin name, or something that's Latinized. And so those requirements, it's kind of like a law, one of these laws, if you like. Otherwise, it isn't recognised around the world. So we're very happy to talk with people about that kind of stuff. And really, it's up to the scientists they work with. Often the names are descriptive of a feature of the plant or perhaps the place, but it has, the people are honoured with the names of plants as well. So so some of our significant botanists and historical botanists have got lots of species named after them now to honour the work that they've done. So it does happen.
So if people find a new plant, or they think they've found a new plant, what do they do?
We do have an identification service, and people can send it to us. There are also people that might be in their local area that are knowledge, some of the dock staff or some of the regional council staff. If they think it's a new weed, they can be a good port of call.
Yeah.
And some of the other collections around the country also can help them with identification. Increasingly, we're trying to put guides up online as well, so people can have a go at identifying things themselves.
But there's also iNaturalist that you can take a photo of and it goes up online, and that there's this whole citizen science thing and all people on there who will help you identify it. So if you're not sure that you can dig it up and send it off, you can take a photo of it or a few photos of it, and people will help you identify it. So there's quite a community out there.
Yeah.
So getting back to the digitisation, you've got it loaded there. What do people do when they want to get started?
So once you've logged in and you've got into here, you click the Get Started button, which is very conveniently orange, and then an image will load, hopefully, slowly. So this is one of our specimens. We can then zoom in so we can...
So it's already been photographed?
So they've all been photographed. So that's how you're going to be transcribing them, is that from the photographs that our volunteers took of them. So, yeah. So we will zoom in.
It's about 1,000 up, I think, isn't it?
Around about. I've got a few more I need to load on. So I'll just zoom in if I can. It's a bit hard on here. Usually I have my mouse. Anyway, here we are. Zoom in. So there are actually quite a lot of details on here. Yeah. Well, some have a lot of details, some don't have so much, but we need to...
We can show a couple of these, can't we?
Yeah, we've got a few other specimens down here. This is just the first on the list itself in alphabetical order. But we look at this information and go, oh right, it was collected from Mount Egmont. So I will go down here into the template and type in Mount Egmont as it's there. So we try, one of our standards, we have database standards, and one of our standards is we have to try and enter the information exactly as it is on the label, which includes spelling mistakes and things, abbreviations, because what the collector is saying, I might guess what it is, but I could be wrong. So we actually want to enter it as the label is, so that other people can work it out, but the information is there.
So in our database systems, we capture that original information, and then we put some other fields, more standardised fields alongside them, which helps people access and find the data and use it as well. So.
Yeah.
So they go through the process and they fill in all
All these details, so
Yeah.
all this information up here, well, some of this information up here. We've put a few things in, like this one is a POA specimen, and we've got a list here. If we go to plant names, it's a drop-down list. And it's got all these names so that you don't have to, there's spelling and things like that. It makes it a lot easier. And so there's things to help you do it, and you can always ask questions on the forums and things like that.
So show us some of these examples. They look pretty cool.
Yeah, they are pretty cool.
So we'll
I'll just move this out of the way.
So we'll just go through some reasonably rapidly. Just show you some more of the diversity in the collection. How's that? Is it about? So this is one of the ferns. And you may be able to see up on here. We've got the back and the front of the leaf, so you can actually see all the spores in the fern. What's Kate giving me now? This one's pretty cool. This is the largest moss in the world. So normally they're, as you know, mosses, the little things in the cracks of the pavement. This one's much bigger than that. You'd actually mistake it for a pine tree seedling or something sometimes. So it's very cool.
Discovered in 1972.
This one was collected in 1972. Yeah. Yep.
Got another algae.
Yep.
Interested in algae?
So this is one of the seaweeds. Oh, there's a piece on that one.
Oh,
Okay.
no, not good.
No.
Okay, put that one back. Right.
What else can you pass me? Yeah, that
we've
one.
got this one here.
So everyone will probably be familiar with this, Ko-Fi. So common plant through the country, common in gardens. One of the more, I guess, pretty plants, if you like, when it's pressed.
So these two.
Yep. So this is harakiki, so the formium, the flax, and we've got 2 specimens of it because this also shows you what we do when we get really large material. So you can imagine a big flax. How do you put one of those big flowering spikes or the big leaves on it? And so we actually fold the leaves up so we get everything from tip to the base of the leaf, and we will take part of the inflorescence and put it on a sheet so that we, as I said before, it's really important with these that we're getting the range of material for people to look at and use.
Threshing plant?
Why not? So this is one of the grasses, and we hold, as I said before, both exotic plants in our collection as well as natives, and this is one of our wee grasses that's actually on the threatened list. So DOC maintains a list of threatened organisms for New Zealand, and this is one of those grasses that's actually on that threatened list. So in some cases, these specimens, as I said earlier, might be from collected from a location where it doesn't occur anymore. So it helps us understand its former range, and actually sometimes to work out, is this actually a different species or not? And
so with the likes of climate change and the real effects of that happening, something like the Allan Herbarium is just going to be crucial now and in the future.
Yeah, and the work that is underpinned by the collection as well. Just the research which is going to help us understand those impacts. It's going to be, as I said before, it's a piece of our national infrastructure, and it's a taonga.
So how can people get involved if they want to help out, they want to be a part? Is the digitisation, is there any other ways that people can help?
So we actually have volunteers that if they're in the local area, we have a program where the volunteers can come out and help with mounting or other aspects around the collection. We're hoping, so that for us this is the 3rd expedition that we've done. The first one actually was on some very hard handwriting, a historic collection. So this is one that the reason we want to try this process is we're putting a bit more variety up this time. And if it works, we'd love to keep this going and to keep people involved that way. I think the question we had before about if they see something really new they haven't seen in their area before, taking a photo and getting it up online or sending it to one of the scientists can be very helpful as well. So
what will happen once the expedition is finished?
So our data, the data from here goes into a number of different website resources where people can access it, and researchers will come and get it directly from us as well. So we have a website for all that covers the data in all of the collections at Manaaki Whenua, because this isn't the only collection that we hold. We've also got fungal and insect collections, and so there's a website that's called scd.landcareresearch.co.nz where all the data is available there. But it doesn't stop there. So there's an Australasian virtual herbarium where we combine all of the data from the New Zealand herbaria with the Australian herbaria, and that comes up in a portal called the Australasian virtual herbarium. That's really important because we hold material from overseas collections as well, and they hold material of New Zealand specimens, and so to get a full picture, having all of that world data up online is important. And then there's global initiatives where the data is available too. There's a portal called GBIF, GBIF.org, and we're one of the partners in that, trying to promote access to this type of data globally.
Cool. Where do people go to find out more information just about the Allan Herbarium?
So we do have a web page that they can find under the collections on the Manaaki Whenua website itself. That's probably the best place to start, and there's some contact and some basic information about the collection from there. And about this expedition itself, I believe the link will go up after we've finished talking here about to the expedition, so you can actually jump on there much more easily.
Yeah.
So I hope that's been really interesting, a little bit of an insight to the herbarium here. I hope you join us online and help us with the task ahead. We've certainly got a huge amount to do. So thank you.
All help is appreciated.
It certainly is.